would now like to welcome Dr. Naveen Vankadari from Monash University, who will be discussing molecular interplay between SARS-CoV-2 and human proteins for viral activation and entry, potential drugs, and scope for new therapeutics. Take it away.
Hi, everyone, and hope you all can see my screen pretty well.
We can, thank you.
Great. Foremost, thanks for giving an opportunity to discuss with it's such kind of a diverse platform and interacting with all over the world. Today, I'm going to discuss about the structurally or functionally how the virus or SARS-CoV-2 is going to enter or engages different sets of proteins of human to establish and enter into the human cells. Let me start with a very a basic thing. Like we all know that the SARS-CoV-2 started from animal source, like in potentially bats or pangolin, and it has spread worldwide, and we're all looking for a potential therapeutics. Well, we are already in the stage of a vaccine, and still it is very imperative to understand how the molecular mechanism of the viral entry is taking place. So that will help you to develop further therapeutics, not only for this corona vaccine, but also in the future any outbreaks comes into. That's why understanding the full repertoire of the proteins and how the cascade of the viral entry mechanism takes place is very crucial. We all know that in very simple and way to explain the virus attaches or enter the cell via a receptor or endocytosis, and then use a host machinery and makes more copies and infect the other cell. That's a very simple way to present the viral replication. But if you look at the complete holistic view, it's much more complex, especially in concern to the SARS-CoV-2 or any other virus. Is not just a single entry mechanism. SARS-CoV-2 does engage a number of host proteins to interact with the host cell and also to enter or engulf into the host cell for the further replication or translation and further processing. We all know and very well familiar because we are hearing that ACE2, that's one of the key receptor which is going to have playing a key role for viral entry. But honestly, there is no clear evidence that if you remove the ACE2, we don't have a virus. In another way, like you can say that ACE2 is one of the main door for the viral entry. The virus can enter with many doors, like in a house with many windows. The thief can enter from a main door, a window, or anywhere. So in similar way, the virus can enter through ACE2 or other receptors or other processing or by a direct endocytosis or a membrane fusion. Many mechanisms are there. So despite you are blocking an ACE2 as one of the key receptor binding, definitely will reduce the viral entry, but you cannot abolish the viral entry in 100% way. So today I'm going to discuss about what are the other three strategic key receptors and the processing enzymes of host, which are making viral entry successful. If you look in normal TEM or SEM micrograph, is not just one virus enter the whole cell as shown in most of the textbook diagrams, like virus enters in one cell and goes on process. Hundreds and thousands of viruses infect in a single cell. And even one virus entry can also succeeded by the number of viruses. Like you can see one endosome contains tens and 20s of viruses. It's not just one virus receives one receptor and then that's only the end of the one viral entry. It's very complex and matter. Today, going to explain how the SARS-CoV-2 engages the three key receptors or processing enzymes or host components called furin, TMPRSS2, and ACE2 and CD26. The first and foremost idea came through our understanding is to unravel what are the key structural and sequence changes between the SARS-1 and SARS-2. I believe we are the first to tell the key differences between the two viruses. We published this article in February and showing what are the structural differences between SARS-1 and SARS-2 in open and closed conformation, just through a computational approach and molecular dynamics, and clearly showing in a key distinct regions like an N-terminal and also the furin binding site, which has been inserted in SARS-1 and 2. There was a key change that's been discovered first. But we all came to know in lately, in probably in April, end of March or April, the SARS interaction with the ACE2 has been structurally shown that the key entity for the viral interaction. But if you look at the SARS spike protein is in 3 dimension, it is very complex and it has many regions has conserved with to have interaction with the SARS-1 and SARS-2 as conservative regions for viral entry. In similar way, in the later results for the cryo-electron tomography, and our colleagues and other collaborators shown that very clear in cell, the cryo-electron tomography shows that the distinct features of spike protein. One is in a perfusion conformation where the SARS virus infect any host cell, or before infection, or while interacting within a human ACE2 or other receptor. It shows in a very distinct conformational features. So, which also says that the ligand binding or receptor binding region of ACE2 can be flexible or very prominent to change. So this also raised in a speculation, and many people has confirmed that not just one receptor for an SARS-2, but there are many receptors can have an importance for viral entry, but has a different potential for viral entry. So based on this, we first saw the structure of SARS-2 in complex with the CD26 is a human fibronectin receptor-based viral entry. So before that, and we've also first showed that how the SARS-1 and SARS-2 has a difference in the glycosylation pattern. Many people ignore and show the several experimental results showing that SARS protein they purified, ACE2 they purified, and show it interacts. But how true it is in realistic way. Most of the human proteins are the viral proteins, when they enter into the cell, they get glycosylated. That's why it's called a spike glycoprotein, or many other human proteins get glycosylation. So post glycosylation and before glycosylation, there is a hell lot of an immunogenicity, and the region where the glycan is binding is very hard to say that place the antibodies can effectively work or receptors can effectively work. So first we have to map what is an effective region of SARS-1 and SARS-2, the difference in the glycosylation pattern, which is also considered as a camouflage pattern of SARS-1 and SARS-2, and showing in it. And first we discovered that what are the 2 regions of the glycosylation patterns, and that helps to identify the non-glycosylation regions where we can use an antibody therapy. Of course, this wasn't very long before in March, so now vaccines are there. But understanding the mechanism was important. That's what I'm going to explain here. And then we have solved the structure of the SARS-2 in complex with the CD26, showing it's a reasonably good affinity in interaction with the, most of the arginine and the lysine residues, and as well as other key regions which are going to have a play important role. Of course, its affinity is way lower compared to ACE2, but it does have an affinity towards an CD26, which is also called as DPP4 or dipeptidyl peptidase. So with that, our understanding of a CD26 with the COVID-19, within around 7-8 months, we received several citations of near 300 citations we received on finding of the SARS-2 interaction with the DPP4, and companies has came through to make a mouse models for making ACE2 and DPP4 mouse models for generating many functional studies and in vivo studies as well. And which also lately came to know that vitamin D has a direct role in the coronaviral replication and infection rate. And of course, the DPP4 is a direct receptor of the vitamin D. So the link continues to go. So as I mentioned in the spike protein, as we know, is a homotrimer. So we are looking for a potential therapeutics which can break the homotrimer of the spike protein. So we first draw an attention that unlike in most other coronaviruses, in the influenza virus, H3N2 spike protein has a conserved region wherein a drug called Arbidol can bind effectively. Then with the set of molecular dynamics and computational approach, we first tested the efficiency of this drug binding to a spike protein of SARS-CoV-2. It does binds effectively. Then we tested with its binding affinity of the drug with the spike protein, and also tested this cytotoxicity in cell lines. So compared to other drugs like a Bolaxir, Lemonafir, and Zemafir, and the Arbidol binds effectively. Of course, it has shown some cytotoxicity in cell lines, but that need to be taken care of. But this Arbidol drug is an FDA-approved drug in some countries, and some countries not approved, and dilemma is there between the drugs. And we also saw the structure of the Arbidol binding to the spike glycoprotein and make the, and we can clearly see the Arbidol binding interface between the two spike proteins where it completely breaks the dimer interface. The breaking the dimer interface between the spike proteins will weaken the structure of the spike protein and thus makes the virus protein least effectively replicate or attach the human cells. In pictorial way to show the spike protein binds in the deeper groove and breaks the interaction between the two monomers of the spike protein. That's how it works as well. And with this analogy and knowing the structure and other, we collaborate with the first and designated hospital in China, that is Wuhan Pulmonary Hospital, and to know how this drug can behave in patients and to test, because that was in pretty early time. And in Australia, we still not have an approval for the Arbidol drug, and we tested for in Wuhan Hospital around 2,000 patients. And we screened down to 500 patients at the last, and seen that in just glimpse, I'm showing in the raw picture, that a patient when admitted with a lung infection, you can completely see the lesion. And after around 10 days, this is the 9th Feb and 19th Feb, you can see that in 10 days, the lesion is completely gone, is showing in a very promising and effective treatment.
You have 2 minutes left of your presentation.
Thank you very much. We also tested in parallel with other combination of the drugs and also an Arbidol alone, and it shows a very clear efficiency to clearance of the virus. That gives a very confidence that Arbidol drugs not only binds to a spike protein, but also clear the virus in patients. So this makes also its odd ratios is in the mortality ratio also being decreased compared to just patients given with oxygen and vitamins. I just skip the slide. The another one component I would like to just say and then end the story, that we all know that the human SARS-2 has a new insertion of a furin protease. And we all know that furin protease cleaves the spike protein into S1 and S2 domains. So if the SARS protein is going to cleave from S1 and S2 domain, where the S1 domain is going to interact with the ACE2, then what is the function of the ACE2? So because the SARS-2 is, the S2 domain is just standing back and makes it functionally inactive and useless for ACE2 present. And we also saw the structure of the furin in complex with the spike protein, showing that how furin recognizes spike protein and interacts, and showing that how the uncleaved and cleaved fragment showing a structurally instability happens when the spike furin cleaves the spike protein of the SARS-2. So this raises, sorry for that. This raises our understanding how the host proteins are going to play an important role in the viral adhesion and entry. Thank you very much.
Thank you for your presentation. So questions will be coming up in the Q&A chat feature. So
Yep,
you can type an answer to that as they come up.
No, thank you.
I have a quick question. Have I got time to ask, Leah?
Yes, you do. Thank you.
Okay, so thank you. That was a great presentation.
Thanks,
my
man.
question to you is, the Arbidol data that you've generated is quite interesting, given sort of some prior data which suggests it's actually not particularly effective. So that's interesting. And I guess what I wanted to ask you to comment on for both DPP4 and targeting and Arbidol.
Yep.
We've seen with the monoclonal antibodies that the challenge has been that the virus mutates. Do you foresee any potential problems with those targets?
I saw that they are not much playing any important role. The affinity between the receptors changes because most of the mutations, either the UK or South African mutation, is a single point mutation. A 1 point mutation usually may not abolish complete interaction within a protein, but definitely changes in either increase or decrease the affinity between the receptor. And if they're making a monoclonal antibody in so particular way that targets only the mutatable region, then actually that antibody is kind of useless. When they have to find a right antibody or even the right region, they have to make a mutation or either the vaccine or the antibody. They have to screen for non-mutable region. For example, in poliovirus, since around 30-40 years, what they made a mutation in RNA still looks fine even today. That's how you have to find the right region where it is non-mutable.
Thank you.
Okay, so that currently concludes our questions. You can just keep an eye on that Q&A section.
Sure, great.